Okay, so basically we left off with the mitochondria. So let's go back to the mitochondria and let's figure out what we're doing. So the mitochondria, most of you guys who have had biology before have heard mitochondria is the powerhouse of the cell. Mitochondria is found in every single type of eukaryotic cell. So plant cells have mitochondria, yes they do. Animal cells have mitochondria as well. That's what it means by every single type. So just make sure that you remember that for the test. Mitochondria is a membrane-bound organelle, and what that means is that it is surrounded by a membrane system, kind of like the nucleus. Also, you have to remember that it actually is a double membrane system, which is really important to remember. There's an inner membrane and then there's an outer membrane, and that creates intermembrane space, which is going to be very important for respiration. And there's also an area called the matrix, and we'll go into detail when we get to respiration. So the main thing is it's covered by double membrane. Another important thing is that there are proteins within that double membrane that basically carry out oxidative metabolism, and all that means is that the process of cellular respiration occurs within the mitochondria. Can't see at the bottom of the slide, which is okay, but it basically says that mitochondria has its own DNA. Now, as I mentioned in class, this DNA is circular. So, hint, hint, think of what kind of an organism has a circular plasmid DNA and maybe doesn't have any membrane systems. Okay, let's think about that. So here's our mitochondria. Here is the cristae. This is the fold of the one membrane. Here's the outer membrane. We have the intermembrane space. Again, remember this is going to be important for respiration. And then inside, this is where the matrix is. Again, all of this is important for respiration. And right in here, these blue and red dots, those are proteins which are stuck in the membrane. Again, going to be very important for respiration. Next are the chloroplasts. Chloroplasts are found in plants as well as photosynthetic microorganisms. Those are very important for the process of photosynthesis. So what that means is inside there is a pigment, think of that as a liquid, which is colored green, and it is really going to be important for photosynthesis. We'll get to photosynthesis within the next week and a half, 2 weeks, so don't worry, we'll go through that. But the main thing to remember right now is chloroplasts are found in plant or plant-like organisms. Chlorophyll is inside. It is surrounded by a double membrane, kind of like the mitochondria. In fact, exactly like the mitochondria. Part of the structure is that thylakoids are membranous sacs. You don't need to remember that for right now. And then grana are stacks of thylakoids. So just picture a stack of pancakes. So the thylakoids are the pancakes and the grana is the entire stack. Again, this is going to be important for photosynthesis. For right now, don't worry so much about the actual structure. And again, chloroplasts have their own DNA. It is circular DNA. It looks kind of like prokaryotic DNA, which is important. So here is our double membrane system. Here's the inner membrane. Here's the outer membrane. This is the interspace. Here are our pancakes, right? So we have our thylakoids, the entire stack. And the entire stack is a granum. Multiple stacks are grana. And then this liquid environment is the stroma. This will be very important for photosynthesis. We will cover this again in a couple of chapters. So I kept on saying things like there's a double membrane system, there is circular DNA that kind of resembles prokaryotes. So here is the endosymbiosis theory. Yes, this will be on the test. Yes, this is very important. If you don't understand how this works, please remind me in class or after class and I will go through this with you. So basically, here the entire theory is based on the fact that we believe, based on evidence, of course, because this is science, based on evidence, it is proposed that chloroplasts and mitochondria used to be single-celled organisms that used to be free-living. And then along came a larger organism, and the mitochondria and chloroplasts, depending on which organism, decided to live within this organism. Now, mind you, decided they don't have a brain, but it must have been advantageous. So these organisms basically let's just say they were engulfed, they became a part of that cell, and then eventually they got stuck. Which makes sense why there's a double membrane system, because they would have a single membrane around themselves, and as they're coming through the outer membrane, the cell membrane, you will have to put this organism into a vesicle. So you have a double membrane. The DNA is circular, again, because prokaryotes have very basic circular DNA. So mitochondria and chloroplasts are both part of the endosymbiosis theory. Okay, so far we have gone through membrane systems. Membrane systems are Golgi, rough ER, smooth ER, lysosomes. We technically have nuclear envelope. We also went through organelles. Yes, this entire structure is organelles, but in terms of true organelles, let's say the nucleus is an organelle. Let's say mitochondria and chloroplasts are organelles. Ribosomes are organelles. Now we're moving on to what I call the third category of cellular structure, and that's the cytoskeleton. If you remember that video I showed you in class, the short video, where you have a protein carrying a vesicle, it is walking along microtubules. So it's walking along those railroad tracks, which is part of the cytoskeleton. So basically, if you picture your cell and you picture the shape of your cell, it is made up of different cytoskeletal components. So if you think of it as a network of protein fibers that are found in all eukaryotic cells, just think of it as a railroad track that keeps the cell in its round, basically circular shape so it doesn't fold in on itself. So a cytoskeleton is important for support, and it also keeps the organelles in their fixed location. This way your nucleus is not just floating around, your mitochondria is not just floating around the cell, everything is where it needs to be. The other thing to remember here is that in microorganisms, and even our cells actually, when the cells are moving, the cytoskeleton keeps on disassembling and reassembling, and that's what we mean by dynamic system, right? The cytoskeleton has to disassemble and reassemble in order for the cell to stay flexible. So that's really, really important. There are three types of fibers. The first type is microfilaments. They're made up of actin. Actin is a type of a protein. And basically, there are multiple proteins, two different types, that connect together. And this type of a filament is important for things like contraction, crawling, and pinching. So when you picture microorganisms like amoeba, the ones that move with the pseudopodia, that crawling is possible because the microfilaments keep on breaking and reforming and basically rolling the cell. Just keeps on rolling and rolling the cell round and round until it gets to where it needs to go. When you think of microtubules, think of mitosis and meiosis. So basically, think of it as these ropes that assemble and disassemble, and their job is to move cell material, especially chromosomes, from one place in the cell to another. Um, it's made up of alpha and beta tubulin. Again, those are proteins. You don't have to know that. But the biggest takeaway thing for microtubules is that they're very important for moving things within the cell, especially chromosomes. And then there are intermediate filaments, which are basically not as big as microfilaments, but they're not as, not as big as microtubules, but they're not as small as microfilaments. And they're basically actin filaments. So again, that's the protein, and they are not broken down. So they just keep the cytoskeleton in the shape that it needs to in the areas that are not normally disassembled and reassembled. This is just a visual. So here are microtubules. Think of mitosis and meiosis. Intermediate filaments, this looks like it's more or less in muscle. And then these are the actin filaments that we mentioned on the previous page. So those are your microfilaments. And as you can see, the the rough ER is basically stuck where it needs to be. The mitochondria is stuck where it needs to be. So everything is where it's supposed to be without just free floating in the cell. Okay, so centrosomes, think of centrioles in microorganisms. Microtubules come out of this. This will be important for when we're doing mitosis and meiosis. So just for right now, when you think of centrosomes, this will not be on the test. When you think of centrosomes, just think of mitosis and meiosis and cell division, and then you'll be okay. Um, Eukaryotic cells have walls. Some of them do. So many plants and fungi and plant-like protists, they will have the cell wall. As you can see, there's the primary cell wall, there's the secondary cell wall, and don't forget, there's also the plasma membrane. Every single organism has that plasma membrane. Depending on whether it is a plant or a plant-like protist, in those cases, cellulose. Now, cellulose, remember, is a macromolecule of carbohydrates. It is made up of glucose, and that makes up the cell wall. In fungi, there's something called chitin. And remember how I mentioned chitin is, again, a type of sugar? When you step on a bug and it goes crunch, that's because there's chitin in its exoskeleton, the outer covering. Um, and that's pretty much what you have to know. And then towards the end, again, this is a little cut off, I'm sorry, cells need to be connected to each other, and cells also need to basically communicate. So if you want two cells to be connected together without anything passing through from one cell to another, so no leakage, you will have tight junctions. So pretty much how the name sounds. If you want to have mechanical attachment of the cytoskeleton to the cells and keep everything in their place, you're going to have anchoring junctions. So you're basically putting down an anchor and keeping everything in the same place. And then if you actually want cells to talk to each other, you will have communicating junctions. So there will be pores, either gap junctions or plasmodesmata, those are the pores, and you can have either chemical or electrical signal being passed from one cell to another. So for this slide, just keep in mind there are three types of connections depending on what you want your cells to do. So this is your tight junction, nothing's getting through here. This is our anchoring junction, so it's connecting the cell membrane to these different protein structures, these intercellular space components. I'm sorry, intercellular spaces right here, to these intermediate filaments components. And then there are other connections where you basically have these gaps, so you can have a chemical or an electrical signal passing from one cell to another, which can be important, especially if one cell is trying to influence another cell to secrete, meaning let out different proteins and help it out with something. So that is it. Good luck. If you have any questions, let me know.